which is studying nausea. Even though nausea is frequently experienced by humans and other animals, our understanding of that underlying neural mechanism remains very poor. Dr. Lee is changing that. I'm sure you will enjoy his talk and learn a few things as well. And there will be time at the end for questions. So any questions you might have, please type them into the Q&A section at the bottom, and we will try to get to as many as possible. Now let me turn it over to Dr. Lee.
Thank you, Terry, for that introduction. It's a pleasure to be here. I thought perhaps it would have been better to be in person, but given the snow we got this morning, maybe it's a good thing that we're having this online. So I'll start with a bit of an introduction to myself. Let's see. Okay. So I'm, as Terry said in her introduction, I'm an assistant professor in the Department of Neurobiology at Northwestern University. And I've been teaching students at Northwestern since 2019. And the other part of my job is to run a research group. I have 9 trainees in my group, and my lab primarily studies the sense of taste. So I'm going to show you this video recorded of children tasting something for the very first time in their lives. So this boy is tasting anchovy for the first time, and this is the response he's showing. And the next boy will be tasting lemon. And as you can see here, the sense of taste lets us identify the good things, the essential nutrients like proteins, carbohydrates, or salts in our food, and also to detect bad things like the sourness of spoiled foods or unripe fruits, or toxins are detected by the sense of bitter. So you don't really have to be a neuroscientist to identify which of these 3 children liked what they tasted and which of them didn't. This is because the sense of taste is hardwired and programmed to elicit very strong responses, stereotype behaviors such as grimacing. You can see that this boy, just by the facial expression is showing, we could even recognize that he might be tasting something very sour. So you might wonder why a taste researcher submitted a grant to the BRF to study nausea. And that's because my lab is primarily interested in understanding the brain circuits that are involved in how taste and various senses translate into these reflexive prototypical behaviors. And one of the behaviors that we got interested in was by looking at how people or animals might respond to something very bitter, where they might start gagging, retching, or even vomiting. So we got interested in the vomit circuit, and this is part of a nausea circuit, which is not well understood. And the particular nausea, so there are many ways to get nauseous, like getting food poisoning, getting sick from medications, or something that I am prone to get, which is motion sickness. So about 30% of the population experience severe motion sickness. However, not much was known about how this is processed in the brain and how the brain makes us feel nauseous. And motion sickness is a form of nausea that doesn't happen from the viscera. It actually is processed within the brain. And what we found to be quite revealing about motion sickness and its connection to nausea is that the word nausea originates from the Greek word for sea. And we can look at texts back from the ancient Greeks, like Hippocrates talked about how sailing on the sea, or nous, proves that motion disorders the body. So even though we've been aware of motion sickness and nausea for thousands of years, our understanding of this within the brain is still quite primitive. The diagram I'm showing you here is from 1978, where reason um, formulated this idea that nausea, motion sickness comes from a conflict of senses where your eyes might be telling you something, but your sense of balance is telling you something else. And when there's that mismatch, that the body responds as being nauseous. So because not much had been done since 1978 to understand this at a cellular or molecular level, we were interested in studying this. And because it's difficult to study the human brain and to get detailed information about how the brain is processing motion sickness, we first had to start by developing an animal model to study motion sickness. And we our favorite animal model is to use mice. There are many reasons for using mice as a proxy to understand how the mammalian brain works. And because we couldn't really put mice on a boat and get them seasickness, we put them on a different device that we developed, which sort of gave them a similar experience to what this woman might have been experiencing. And we can see that this animal uh, got motion sickness by the fact that they weren't moving very much after this. Once they feel motion sickness, they also show other signs that they're feeling sick, like squinty eyes, stiff tail, and the fur all standing up. So having developed this primitive behavioral assessment of motion sickness, we went further and also identified that motion sick mice and many other animals actually experience chills. It's like many of us when we experience um, car sickness, motion sickness, or even other forms of nausea, and we're about to vomit, we commonly experience these chills. And we were able to detect that in our animals after giving them motion sickness when we use an infrared sensor, temperature sensor. We could see that before before the motion sickness, these animals' skin temperature was about 32 degrees Celsius. During the shaking, their temperature goes down to about 27 degrees. And then once they recover, they go back to their normal skin temperature of 32 degrees. We can assess this more precisely using a implantable probe, temperature probe. And by doing that, we saw that control animals maintain their body temperature, but once they're subjected to motion sickness on that shaking device, their body temperature drops almost five degrees Celsius. So that this was a very reliable way of assessing motion sickness in mice. So what do we want to do with that? As neuroscientists, we first wanted to understand which part of the mouse brain is involved in initiating that sense of motion sickness and to translate that to hypothermia and other vomiting responses and other kinds of phenotypes, behaviors. So we first started by looking where in the mouse brain is this encoded. And we saw that when we looked throughout the mice of the brain of mice experiencing motion sickness, we saw this area of the brainstem which were activated. So this is a mouse brain which has been made transparent, and the active parts of the brain are labeled with brightness or white. And you can see that there's a lot of brightness here. And this actually translates well to what was known in the literature but had been ignored. So in 1951, this exact same area had been proposed as the vomiting center. So this gave us confidence that we might be looking in the right places. But 2026 is such an exciting time to be a neuroscientist. There have been many great tools that have been developed over the past decade or so that allowed us to, for example, this image on the left is something that would have been impossible to do, to clear the entire mouse brain and look at the precise areas of activation. There are other tools that have been developed that helps us understand how the brain works. And one method is a way of going back to these actual neurons and artificially being able to stimulate them in live animals. So when we do that and we stimulate what we think are nausea neurons, we and that's at this point, we could recreate that nausea behavior, even though we're not putting them on shakers or we're not doing anything else to them. We're just artificially stimulating these handful of cells very selectively. And by doing that, um, we were able to see that once you activate those nausea neurons, the body temperature of that animal drops just as if they have experienced that motion sickness from shaking. So you see that the body temperature drops quite significantly. So this drop, you can see the drop coming from like almost 36 degrees centigrade, which is the normal body temperature, to, and it goes all the way down to about 28 degrees Celsius. Uh, and and I'm sure that when you experience nausea, that you also one of the behaviors that you show, or one of the responses that you show, is that you don't want to eat. So we tested that in these same animals, where we artificially stimulated the nausea neurons. And of course, these were done in hungry animals where they're eager to eat. So these hungry animals would eat food when given presented with food, but by activating those nausea neurons, we saw that they no longer were interested in eating. Actually, I wish I had a video of this, but it was kind of cute. We saw that these animals are feeling nauseous but interested in food, so they would go close to the food, maybe and sit on the pellets, but not show any interest in actually consuming food. So that really summarizes what we were able to discover in those 2 years of my seed grant. Um, but of course, this has many interesting and important outcomes for the future. We're still interested in understanding the basic biology, the fundamentals of science of how the brain detects and causes nausea. We think that nausea is a way for the body to tell the brain that something is wrong and something has to be done to prevent that. And that's why we think that with motion sickness, when the brain is detecting that the the outside world is not behaving in the way that it's expected, that the eyes and the balance doesn't match, that they have to tell that animal, the brain has to inform that animal to stay put, be immobile, and just shelter it out until things get back to normal. So we're trying to understand the circuits, the neurons that are involved in this process. Of course, nausea, as I alluded to earlier, is a very common complaint from people. There are many factors that can cause nausea, like motion sickness, pregnancy, many illnesses, side effects of many medications. And we're hoping that whatever we find in our studies, in our research, will help in the development of precise therapeutic interventions. Even if you look in the literature these days, they would say, oh, drink ginger tea to help with your nauseousness. So developing precise ways to target sickness, vomiting, morning sickness would really help um, many, many people. And in addition, we found that um, by activating nausea neurons, we are changing blood pressure, and that's what causes the temperature to drop. So in a way, this work has exposed us to yet another avenue of research. So I started off as a case researcher. I moved on to this understanding nausea now, and this um, would have, would not have been possible without the generous support of BRF's seed grant, which I received from 2023 to 2024. And besides these outcomes from the science side, from the research side, there are many tangible outcomes that came from getting this seed grant. One of which was that, as many of you may know, Northwestern University had our federal funding frozen for about a year, and that this was resolved only quite recently, back in December. But during the times when we couldn't get any access to federal funding, private foundations and, for me personally, BRF money helped me and my research lab stay afloat. But maybe to end this on a even more positive note, um, that 80,000 BRF seed grant translated into me my research proposal to NIH being selected. So last year I was awarded this grant, 5-year grant, major grant from the NIH, which will give us about $3 million of support. So um, I would like to use this platform to thank all the philanthropists in the audience who make science possible, and hoping that your generosity translates to more discoveries in science. With that, I would like to thank you for your attention. I might just want to point out that the bulk of the research that I showed you today was done by a fantastic graduate student in my lab, Alex Peckerman. This is my lab here. You might notice that we all are wearing these white patches, which are actually temperature sensors. When Alex discovered that mice experience chills to motion sickness, we took that as an opportunity to maybe have a lab outing. So we all wore temperature patches and went to Six Flags to get on some roller coasters. Anyways, thank you so much for your attention. I'll be happy to answer any questions.
Well, thank you so much for that, Dr. Lee. I'll remind anybody, if you have a question, please type it into the Q&A below, but I have a couple that are there already, so I wanted
Mm.
to start with those. So, Dr. Lee, you mentioned pregnancy before, and one person is asking, why are some mothers affected by nausea during pregnancy while others are
Mhm.
not?
Yes, we're still trying to understand that. And perhaps that's an opportunity for me to point out the importance of doing animal research. In humans, there's a lot of variability between people. Some experience severe morning sickness, some people have no issues. And one of the benefits of using experimental animals like mice, which are bred to be genetically identical, you get the same genetic background, so you don't see individual variability as much. So that helps us in understanding which brain regions are really important in morning sickness. So even though my lab we haven't really gone into pregnancy, I think that's something that we can explore.
Okay, thank you. We have another question, and they are asking a little bit more about the connection between nausea and blood pressure.
Oh, and so the nausea neurons that I mentioned are looking in the old literature, we found that these neurons um, are where the projections from the cardiovascular system, the sensory neurons from the cardiovascular system uh, projects onto the nausea neurons, or what we suspect are nausea neurons. So we're exploring that link between the cardiovascular system and the nausea neurons.
Okay, we have another good question, and they're asking about, are there any certain foods that people can eat to make the brain perform the best?
Oh,
You had mentioned, I guess,
I
ginger, right? Yeah.
right, yeah, yes. I think that's a good question. Uh, I don't have ideas for specific foods that we can eat, but as a taste researcher, I think it's important to um, eat very diverse things broadly, and not to eat in individual items too much. So diversity, I think, there of getting different sorts of nutrients is important.
Okay, another person is saying you stimulated the nausea neurons. Is there a way you can do the opposite
Abs-
to stop nausea?
Absolutely, absolutely. And although I didn't show that result here, we have also um, silenced those neurons
Mhm.
temporarily, and we saw that in when you silence those nausea neurons, the mice that we experimented with do not show severe signs of motion sickness. So their body temperature, in spite of them going through the motion stimulus, they do not, their body temperatures do not drop as much as the regular mice. So yeah, we have shown that.
And this is a follow-up question. I think it's kind of on the same vein. What can we do? Is there something, what's your ultimate goal? It seems like you can manipulate this in animals. How can we bring that to humans?
Yes, absolutely. I think because this is so prevalent in the human in our population, uh, nausea as a whole, but even motion sickness. Again, about 30% of the population experience quite severe um, motion sickness. So we're hoping to hone in on the molecules and perhaps the cells that are involved in this process and contribute to developing therapeutic strategies.
Okay, people really like the temperature and blood pressure changes. They have a lot of questions on that. So here's a question. Do you think the temperature drop is casual or to the nausea or just is causal to the
Causal,
nausea
I
or
see. Yeah.
just co-occurring?
We're not sure yet, but we're exploring that. Yeah. We would like to understand what comes first. Is it the temperature drop that causes nausea, or is it the nausea that causes temperature drop? So we're trying to answer those questions this very
Oh,
moment.
and a follow-up kind of question. The temperature and blood pressure changes in motion sickness, are they mediated by the autonomic nervous system?
Absolutely. So the part of the brain that we're looking at is well known to be involved in the autonomic nervous system. It's part of the parasympathetic pathway. So we believe that's why it has such a strong impact on the cardiovascular system and the body temperature. We just have to tease apart where in the circuitry these cells actually lie. Um, Perhaps something that I didn't really go into detail of is that for motion sickness to happen, there are many steps. So there has to be the detection of the mismatch. There has to be the calculation of it. There has to be all the responses that we see, the body temperature drop, the immobility. So we're trying to figure out where our nausea neurons lie within that whole pathway.
I have a follow-up question about the patches that were on your lab. So did they work? Did they work? And were certain people, more susceptible to the roller
The,
coasters and the motion
w-
sickness?
with, Without making this too inappropriate, it was a very warm day.
Okay.
So um, the the temperature fluctuations we saw were mostly on whether we were standing in the shade or in the sun. So I think there could be more invasive ways of precisely measuring body
Okay.
temperature, but we're not ready to go there yet.
Okay, or go on a cooler day, Six Flags
Yeah,
on a
right.
cooler day. Okay. And then I see one more question, and
Okay.
I actually of having this happen to me too. I didn't have motion sickness or feel nauseous when I was doing things when I was younger, but as
Mm,
I get older, I seem to it's be affected a little bit more. Is that
mm.
common? Is that, why? Why would that be happening?
Again, I think that really speaks to the differences between individuals and the state that they're in. Maybe age is a factor, gender might be a factor, and I would like to emphasize the importance of doing animal research because that gave us the ability to compare these different factors. And in genetically identical animals, we've tested our motion sickness protocol on young mice, old mice, female mice, male mice, hungry mice, mice that had just been fed. And we see that the stimulus that we're giving is very reliable and consistent across animals.
Okay, great. I was gonna follow up with, do you try it on male and female?
Yeah, yeah,
So,
yeah.
great.
We don't
Uh,
see great differences between them.
I'm sorry, you do or do not?
We do not.
Oh,
So
interesting.
I think this is a universal stimulus that and a universal response.
Okay, very interesting. All right, I don't have any other questions.
Yeah.
So I just want to say thank you again, Dr. Lee, for that interesting talk. And I want to thank everyone for joining us today. Be on
Mhm.
the lookout for another lunchtime lecture announcement. We're going to start doing these more regularly, and we hope you will join us again to learn more about the interesting neuroscience Brain Research Foundation supports.
Thank you very much.
Thank you.